Good afternoon, everyone. It's a great pleasure to spend some time with you this afternoon to talk to you about the exciting research that's being supported by NINR scientists. So here's an overview of my presentation. I'm going to start with a background. I'm going to give a definition of terms and make sure that we're all on the same page when we're talking about symptom science and precision health. I'm then going to talk about the many aspects of symptom science and precision health from an NINR lens. You're going to be given a tour of the great diversity and variety of what symptom science and precision health research looks like from an NINR lens. And finally, I'll end by sharing with you what I think is a very promising future in this area of research. So let's start with the background. To talk about precision health, we really need to start with a definition of precision medicine, which is as follows. It is an innovative approach to disease prevention and treatment that takes into account individual differences in people's genes, environments, and lifestyles. This is a quote from President Obama's Precision Medicine Initiative that was first launched in 2015. A critical element of the Precision Medicine Initiative is the All of Us research program. It is, the goal of the All of Us program is to gather data from 1 million individuals to accelerate individualized prevention, treatment, and care for all. Mr. Eric Dishman is the director of the All of Us program, and Dr. Steph Devaney is the deputy director, and she actually will be speaking with us a little bit later this afternoon. So she'll give you a lot more background and information about the All of Us program. I do, though, want to add and emphasize that NINR and the nursing community is very much engaged in the All of Us research program. For example, we are a member of a trans NIH liaison coordinating team, and our role in this effort is to act at the interface between NINR grantees or potential grantees, and the All of Us program. We help to ensure that our institute's interests are taken into account as the All of Us program evolves. Only one of many examples of nurse involvement in the All of Us community All of Us program is the Association of Nurses in AIDS Care, or ANAC. They are one of the All of Us program's community engagement partners. So now let's talk about precision health. So I gave you a sense of what precision medicine is, and now that's the foundation upon which precision health evolved. One of the best definitions of precision health I came across is from this quote from Dr. Usha Menon from the University of Arizona, which I will now read dramatically. In precision health, we're not just talking genetics and genetic testing and their influence with regards to a patient's risk for developing diseases and how a patient responds to medications. We're also considering where a patient works and lives, what they eat, their hobbies and how they play, their culture, and how these social determinants affect their health risks and health goals. Another critical aspect, and when talking about precision health, is the emphasis on optimizing health prevention and tailored interventions. That's a really important piece to the precision health lens. This quote is from Dr. Kathleen Hickey et al., a recent review that they published. So we talked about precision health, talked about precision medicine, and there's also symptom science. So let's define symptom science. That's basic and clinical research on the biological, genetic, and behavioral mechanisms to develop improved, personalized strategies to treat and prevent the adverse symptoms of acute and chronic illness across diverse populations and settings. And so you might be hearing some common themes in the definitions that I shared with you, and that's a really fantastic thing because that aligns well with when you're thinking about symptom science and using precision health approaches. And so to make this a little interactive, because I feel like I'm miles away from all of you, you know, when you think of symptoms, could someone call out a symptom you think of? So that comes to mind. Fatigue, yes. Well, how about one more? Pain. Fantastic, thank you. Exactly. Pain, fatigue, nausea, sleep disturbance, and one that I don't have listed here is public speaking induced anxiety. These are all symptoms that we're all familiar with. I will be giving you examples of how we're studying many of these symptoms and the anxiety induced by public speaking you're witnessing in real time. And so what is a way to integrate the concept of symptom science and the concept of precision health? Well, there is one model available provided by Dr. Kathleen Hickey and other NINR-supported researchers, where the foundation of this model is based on the NIH symptom science model developed by Dr. Ann Cashin and others at NINR's intramural research program. The basis of the symptom science model begins with a complex symptom, followed by extensive phenotypic characterization of that symptom or symptom cluster. So that can include gathering clinical data, biological data, behavioral data. These data then are used to inform a biomarker discovery. That would include biological pathways, omics pathways, et cetera. And then findings from biomarker discovery then are taken to clinical application. And so an extension of the symptom science model is the precision health piece. And so you have, for example, precision in measurement. I like to think of this really in the context of how one approaches the downstream characterization of a complex symptom. As shown here, oops, excuse me, I got excited. Let's see, that is a good demonstration of the anxiety we were just discussing earlier. And so precision in phenotypic characterization, so that would include lifestyle, environmental factors, for example. Precision in characterization of genotype and other biomarkers. And finally, which in my opinion is the linchpin or a linchpin of the precision health concept, and that is the personalization of interventions, so targeted interventions and targeted intervention design that would include self-management and intervention delivery. This portion of my presentation, I'm going to give you a whirlwind tour of a lot of exciting research that NINR is supporting. And the purpose here is to show you that there are many, many ways to conduct symptom science precision health research. But first, let me give you a quick lay of the land regarding the grants that we support in this area. And what I really want to call your attention to, this chart shows how the different types of grants that we support. And so the 30% represents the R01 series, which most of you are familiar with. But what I want to draw your attention to, and it aligns very nicely with what Dr. Cashin mentioned earlier, is the large proportion of the grants that are pre-postdoctoral awards. These are individual fellowships that are awarded, and career development awards, such as K's. This is really exciting for us, and it speaks to the promising future that I'll be going into a little bit later. So let's dive in. I'm going to give you an overview of 3 very recently funded grants in our portfolio, and then I will provide a brief overview of 2 published papers by other NINR researchers. So let's start with Susan Westmiller's work, which focuses on the genomic underpinnings for breast cancer treatment-induced nausea and vomiting. The goal here is to characterize the variability of the presentation of TINV in patients treated with breast cancer. Dr. Westmiller wants to take into account several factors that might influence the variability of these symptoms, which include individual factors, as you would expect, age, race, et cetera. Treatment-related factors, very importantly, the type of chemotherapy that the cancer patient undergoes. And then co-occurring symptoms such as sleep fatigue, sleep disturbance, et cetera. But the linchpin here is also an effort to understand whether there's an impact of genetic variability of an individual presenting with these symptoms, and whether or not the genetic variability can tell us something about the variability of TINV. So this could lead to, you can imagine, determining those who are at higher risk for expressing these symptoms more severely and perhaps taking a step towards earlier interventions. The next study is an omics-based intervention to treat pain. Here, the context is functional abdominal pain, gastrointestinal disorders in children. This is work done by Robert Schulman and Rona Levy. I was pausing because I couldn't think of the second PI's name. I believe I have that right. So what's going on here? So children afflicted with FGIDs, research has shown that two interventions that have been tried, cognitive behavioral therapy, CBT, and a particular diet, only works 40 to 50% of the time. That's just not acceptable. And based on other studies done by Schulman's group, separate studies, one showing that biological factors such as heart rate variability, for example, might play a role in how one responds to CBT, and an individual's microbiome makeup might influence how one responds to a diet. And so he's proposing a randomized clinical trial where individuals will be randomized to receive either CBT or the low FODMAP diet, that's spelled out down here if you're interested. And we'll ask, the primary outcome, of course, would be pain at a given time point. And he's hypothesizing, or they are hypothesizing, based on these other data and this finding about responsiveness to CBT and diet, that individuals who do not have what he's calling abnormal markers, biomarkers, if you will, might perform better, might have a better outcome with the CBT intervention, whereas those who do have the abnormal markers might respond better to the diet. So what's exciting here is that here's a possibility that we could use one's microbiome in this particular context and determine which intervention they might have more success with or better outcomes with. So this will be the last study in terms of newly funded grants that I'll be talking about. This one is also, all of these are really exciting, but this one is really exciting too. And so the PIs for this center grant are Gail Melkus and Jacqueline Taylor. Susan Malone is the PI for this pilot project for this P20 award. Okay, that's what I was trying to do. So there's a lot going on here. The study is looking at extending sleep to reverse metabolic syndrome in middle-aged adults. So let me provide some context through this graphic. Metabolic syndrome is a collection of conditions listed here that put individuals at very high risk for cardiovascular diseases, type 2 diabetes. Another piece of this puzzle is the issue of sleep or lack thereof. And we know that insufficient sleep itself has several negative outcomes that can affect a variety of symptoms here and health behaviors or not so healthy behaviors. And so this group posits that sleep duration might in fact be an upstream factor. By influencing these factors can influence metabolic syndrome and symptoms associated therein. And so the study is to use a population, is to study a population of individuals who are community dwelling, racially and ethnically diverse, who will undergo an intervention. And I should also mention, I have to mention this one thing. Let me digress for a second to mention the PI said very wisely that when an individual is given advice to sleep more, what do you do with that? That's not an actionable recommendation. And so they posit that perhaps a particular sleep duration, sleep extension intervention might in fact not only affect sleep duration, so the primary outcome in this study then would be increasing length of sleep, but also would lend to more positive outcomes regarding symptoms of fatigue and more positive outcomes regarding health behaviors. And the other thing that's really important about this study too is that it takes into account the entire socio-ecological environment of the individual. That's the importance of having community dwelling participants in this study and a diversity of individuals in this study. A lot of sleep studies are done in the laboratory, and that laboratory studies do not give you the opportunity to take into consideration all of these other factors, the exosystem, microsystem, individual level, that then feed into individual behaviors that drive sleep duration. The next two studies are published work, one from Dr. Margaret Heidkemper, and this is on microbiome composition and extraintestinal pain in irritable bowel syndrome. And so there are recent data that suggests that alterations in the gut microbiome composition play a role in IBS pathophysiology and clinical symptoms. But what's less well understood is what is the connection between an individual's microbiome and extraintestinal symptoms such as headache, joint pain, muscle pain, etc. And so in this particular study published by Dr. Heidkemper, she was able to demonstrate that, and this graph shows microbial diversity increasing along the y-axis and pain levels increasing along the x-axis. And what she found was that as the microbial diversity increases, the more diverse the microbiome is, the less the experience of extraintestinal pain. And so this is an exciting result because this then suggests that based on one's microbiome, we can get a sense of how likely one might be to experience pain and then intervene accordingly. So my last whirlwind tour of NINR-funded research is another publication. This is by Kanho Zhao. And the context here is cancer-related fatigue. There is a concept known as conserved transcriptional response to adversity. The way it works in general is that individuals who are exposed to prolonged adverse conditions show an increase in transcription of pro-inflammatory factors and a decrease in transcription of antiviral factors. Dr. Zhao's group wanted to ask, do we see a similar profile in individuals who are suffering from cancer treatment-related fatigue and head and neck cancer? And sure enough, that is in fact what they found. At the top, you're looking at the transcription factor profile in fatigue-related, individuals with fatigue-related symptoms, where you see increased expression of the pro-inflammatory genes and decreased expression of antiviral genes. Interestingly, in a group of patients who are also head and neck cancer patients, but they are of the HPV-related variety, whom they had already identified and described as having low fatigue, it turns out that their transcription profile is just the opposite. And so there's a decrease in expression of the pro-inflammatory genes and an increase in expression of antiviral genes. Some of that you can probably expect being an HIV infected, induced, rather, condition. But what's really intriguing, where Dr. Zhao wants to take this work, is to ask whether or not this differential, I'm messing up again, this differential regulation of gene expression might lead to a molecular mechanism that ties fatigue to inflammatory pathway, for example. And what might then be happening for those individuals who do not express heavy fatigue symptoms, and as a reflection of that, possibly show a decrease in expression of pro-inflammatory markers. So I'm going to end talking about the promising future. So I mentioned earlier that slide that showed the 42.5% of the grants that we're funding in this area are by pre- and postdoctoral and early career stage individuals. And so I just wanted to acknowledge a few of those individuals here. Murad Gormley, who's exploring the effects of genomic testing on fear of cancer recurrence among breast cancer survivors. This is an F31, so a predoctoral, individual predoctoral fellowship. Jinbing Bai, who's exploring the microbiome, gut-brain axis, and psychoneurological symptoms in children with solid tumors. This is a K99R00 awardee, which many of you might be familiar with. And finally, Dr. Veronica Barcelona de Mendoza, who's looking at DNA methylation, preterm birth, and blood pressure in African American children. And she has a K01 Career Development Award. And so the future is bright regarding precision health. I've shown you all different types of research that's done in the precision health domain. I focused on symptom science, but this is an approach that could be applicable in a wide variety of different types of science areas. And this is also serves as a reminder of what I said earlier, that in the context of precision health is genetics, culture, even hobbies can help determine a course of treatment. I wanted to almost end, this is my second to last slide, I promise, by providing further evidence of not only the bright future of precision health and symptom science research, but the really critical role that nurses and nurse scientists play in advancing this field. I show here a quote from Dr. Jacqueline Taylor that reads as follows: "Because nurses are at the forefront of patient interactions, it seems only fitting that nurse scientists lead the charge in genomic research that can be translated from bench to bedside." And finally, NINR is leading the way in this effort. I hope that I've convinced you of that in my presentation today. And as I said before, the focus of my talk was on symptom science and precision health, but certainly precision health is applicable to all of the areas of science that we support at NINR and beyond. And so with that, I thank you for your time, and I'm happy to take any questions. Yes, please.
You seem to have a pretty traditional approach to saying that in terms of precision medicine, I'm saying it's genetics. And I was just wondering about other phenotypes. With imaging techniques and that, when we're looking at neurobiologic phenotypes and that. And is it just that nurse scientists aren't doing much of that yet? Or you only had so much time?
Well, I think it's really more the latter. I only had so much time, and you're saying that does make it look like there's a leaning in that direction. I'm going to speculate. I think part of it might have to do kind of with the historical definition of precision medicine and how it included, it was very omics heavy. That might be part of it. But the other part too, and I should also mention that we'll be hearing from Dr. Paul Macy, who is going to be talking very much about neuroimaging. I won't steal his thunder. I'll let him go into that more. But it's not meant to give the impression that it's only omics and genetics based.
I just think that as we train the next generation of nurse scientists, that we broaden our definitions along the way, and I'm sure you embrace that, but I just wanted to make it public here.
No, I appreciate that. Thank you for that feedback.
I was fascinated with when you were talking about conserved transcriptional response to adversity.
Yes.
And I was thinking of the intergenerational transmission of stress, and if anything was done, or we were considering some of the maternal child health work, which is showing like without the poverty and other factors, African American women are more likely to have preterm birth with the best of circumstances, and so that whole area, does it fit there? Because it sounded to me like it might.
I would think it would. I'm not familiar with the research in that area, so I don't know, but I can certainly see how it would be relevant.
It seems like it might provide
Absolutely.
one possible explanation.
Absolutely, yes.
you did a beautiful job. So whatever the anxiety stuff you were displaying, I guess you didn't do very well at all.
Thank you.
But, and I asked this question not to point holes in anything, but to perhaps understand more of the complexity around precision health. And what does the model, or how should or could the model incorporate patient preferences? So their choice to do or not do things, or to enjoy or not enjoy things. And I only saw the physical side of that and not the spiritual sort of holistic side of it that I think is critical to nursing.
It's absolutely critical, and I appreciate your pointing that out. In fact, I was sharing with Dr. Macy right before I gave my presentation that I felt that piece was lacking in my presentation. And it's not meant to, it was not meant to intentionally leave out that very, very important element when we're thinking about precision health. I absolutely would see that in fitting in the phenotypic characterization, for example, where you can ask questions that have to relate to the whole person. I also can see it fitting in at the clinical application, the intervention side, and being mindful of all of those factors that contribute to making the most effective targeted intervention treatment for that individual. So I very much appreciate that point. Thank you.